All right, welcome to Chapter 5, our first section. We're going to be talking today about Section 1, which is Chapter 5. We're going to be talking about the science of heredity or genetics. And today we'll be discussing the work of Gregor Mendel, who was a famous scientist considered the father of genetics and heredity. And so we'll talk about him, and we'll talk about really what he brought to knowledge, what we learned about genetics through him. Two big objectives. One, what were the results of his experiments or his crosses as we'll come to know them? And two, what controls the inheritances of traits in organisms? Those are our two objectives for today. So let's jump right in. First of all, what we need to understand and start with is getting an idea of what genetics actually is. And so there's a couple terms we want to, first of all, come to terms with. The first one is known as heredity. And heredity is, by definition, the passing of physical characteristics from parents to offspring. And so in this little baby down here, the baby has all of these different cells and structures that have been created, and those were passed on from that child's parents. So all the physical characteristics of the child came from the parents. That's heredity. Genetics is just a term that is the scientific study of heredity. Or we could say genetics is the scientific study of the passing of physical characteristics from parents to their offspring. So we study how genes get passed along from one person to another. Now this was founded by this guy over here. This is Gregor Mendel. Then he look. He's got his nice suit and looks very focused for what he wanted to do. Gregor Mendel, known as the father of modern genetics, really famous because he did something that a lot of us wouldn't do. He was patient. He took his time. He was very detailed in his research, and over a long period of time was able to actually study and pea plants and tell us a lot about heredity just through his careful study and scientific experimentation. So heredity, genetics, those are those two terms that we want to work through today. Now here's what Mendel's did. This was his life work. Gregor Mendel used pea plants. Okay, pea plants tend to be self-pollinating plants, which means that they typically don't need multiple plants. They pollinate from one flower and another on the same plant. And what he did is he actually went through and did what's called a cross. He crossed or hybridized the plants. He took two purebred plants, and here's what a purebred is, just to help some of you. A purebred is the offspring of many generations with the same trait. So if this plant had the same trait, let's say flower color, and it was all the same flower color that plant's parents and the parents before that and before that, those would be known as purebreds. We see that term purebred a lot in dog breeds. A purebred dog is a dog that if you go back in his family lineage, that dog has all of the same types of traits or qualities. So a purebred Labrador retriever or a purebred poodle. Okay, so what he did is he took two purebred plants that had different traits, okay, and he crossed them. So he actually, show you how he did this. In step one, what he would do is he would remove the pollen producing structures from a flower. So he'd actually go and he'd cut those pollen producing, those are called the stamen. Okay, we'll learn more about these when we talk about plants. Okay, and he would take those stamen and he would remove them. Okay, then what he would do is then he would take a brush to take the pollen from another plant. So he took the brush from, now notice flower color here. We've got a white plant that's a purebred, and we have a pink flowered plant that was a purebred, okay? And so he'd take the pollen from the white plant, and with a brush, he would brush that pollen right here on the stigma, okay, on the pistil of the flower, that portion of the flower that would be where the pollen would actually adhere and where reproduction starts. Okay, so he would start by clipping off the stamen of this flower that was going to receive, so it couldn't accidentally, the pollen couldn't accidentally pollinate within the flower. He then brushed some of the pollen from one plant to the other. And then finally what he found is then the egg cells that would form, so here's the pea pod, in that flower were fertilized. So we had the egg of the pink flower, right? fertilized by the sperm that are in the pollen, the sperm of the white flower. And what you end up getting were peas in a pod, and ultimately plants that were a product of a purebred white plus a purebred pink. And that's how he could ensure that you didn't have, maybe they self-pollinated and you had an issue. So that's what Gregor Mendel did. Now, here's what Gregor Mendel found. So he did that over and over and over and over and over again. Now, he would have these 3 generations. So in all of his crosses, what he found is only one form of the trait would appear in these different generation terms. Now, we need to figure out some things here. This P generation, you can think of this as the parent generation. Okay, so the parent generation, he would take a tall, let's say the trait he's looking at here is tall. Notice a tall pea plant and a short pea plant, and these would be purebred. That's what he would expect. So you'd have a purebred tall and a purebred short. And then what he would do is he would reproduce and cross those, okay, as we just saw in the last example. And over time, what would happen is he would get that next generation, that first generation of offspring. Those would be known as the F1 generation. Okay, this actually comes from the word filial. Okay, that's the word that we're looking at. And filial is a word that is actually used to refer to, it literally actually comes from a Latin word that means child, okay, or daughter is the direction, the specific answer. Okay, so we're talking about the offspring or the daughters, right, of this parent generation. And here's what he found. Only one form of the trait appeared. Look at this. In the first generation, only the tall plants appeared. There were no short plants. In all of his crosses of a tall and a short, he would always get a tall plant if they were purebred. However, in the second filial generation, so this would be the generation that came from the F1. So if you were to write this out, you had the P generation, right, that then gave offspring to F1, that then gave offspring to F2. So we're actually kind of following down this trait. This would be like grandpa, parents, grandkids. In the F2 generation, he found that, that lost trait, that short trait, would show up about 25% of the time. In 1/4 of the plants, or 25% of the time, you would see and receive this short phenotype, this trait. So it would reappear in generation 2. So that's what he saw. He saw what happened from parent generation to F1 to F2. Now, what he did over lots of careful study and research is he realized through these different traits that there were very specific different traits between one and the other. Okay, so he actually, for example, took the seed shape and the seed color and the seed coat color and the pod shape and the pod color and the flower's position, where the flowers actually arrived on the stem, and the stem height. He took each of these things and he found that, some of them were controlled by what he called a dominant factor. Okay, and that word dominant, think of it as if somebody's dominant in a sport, that means that, man, they're the best. They can win. The other ones were controlled by what he called a recessive factor. And so he said, what he found was that if he took a round and a wrinkled seed-shaped plant, and they were purebreds and they had offspring, they would always be round in the F1 generation. And yellow and gray and all these things would be consistent. And so what he started to realize as he studied is that there were these very distinct traits, okay, that were dominant, that showed up all the time, and other traits that were recessive, that only showed up occasionally or in several generations. Okay, and we'll talk more about this as we progress. Now, what he actually did is he had found genes. Okay, we now call them genes. Mendel called them factors. And he said that these factors controlled a trait. Okay, a gene is all of the different things that we now know on your genetic DNA, on your information, that control your phenotypes, what you look like. Okay, an allele is a different form of a gene. So you have 2 alleles, for example, you have 2 alleles for your eye color, let's say. You have one from mom, one from dad. or 2 alleles for dimples, one for mom, one for dad, or 2 alleles for whether you have, unattached or attached ears. That's what was found by Mendel. He was actually discovering genes and alleles. And what he had found is that we have dominant alleles, and this is where the trait always shows up when it's present. And we have recessive alleles. That's when the trait that is hidden only shows up, okay, when a dominant allele is not around. Okay? So this trait is hidden when a dominant allele is present. It gets masked. Okay, so you won't see it. Now down here in this drawing, I just showed a couple examples of different alleles, right? So here's individual A, who would have, here's an allele for blue eyes and allele for brown eyes. Now if brown is dominant, what color eyes would this person have? They would have brown eyes. Person B, individual B, has a brown allele and a brown allele. They're going to have brown eyes. Person C has a blue allele and a blue allele, and they're going to have blue eyes, right? And this would be, these words right here you see, we'll learn more about these, heterozygous and homozygous. Heterozygous just means the alleles are different. Homozygous means they have the same type of allele. This would be more like purebred is what we'll deal with. So that's what he found with genes and alleles. Now let me help you out with some of the symbols. When we talk about alleles, we're going to use 2 symbols. We're going to use a capital T, okay, and this is just, it can be other letters, but capital in a letter is always going to express a dominant allele. Lowercase will always express a recessive allele, okay? So genotypes, or the genetic makeups of an organism, are either going to be capital T, or lowercase t, or capital T, lowercase t, which could also be expressed as lowercase capital, but it's the same thing. Okay, so down here we have an example. So we have the white rabbit here. The white rabbit, okay, which in this case white would actually be a recessive gene. That rabbit's alleles, we would know, have to be little t, because if he had a big T, he wouldn't express that phenotype. He would actually he would be black. Because a black rabbit, if the big T is dominant, he's either got the genotype big T, or big T, little t. So he's either a hybrid or a purebred organism, but we know that he is going to be black because he has that dominant allele of the big T, and we'll talk a lot more about that in class. Lastly, I just kind of alluded to that, but purebred versus hybrid. Purebred, remember, as we said, has the same traits passed for generations. Purebreds are you're going to see homozygous alleles. Okay, that is huge. Homozygous. Homozygous, remember, as we said, homo meaning same. Homozygous means the same genetic information, the same alleles on that gene that they're coding for. So purebreds have the same traits. They're going to be big T, or little t. If I look down here below, okay, this would be, we don't know for sure if these are purebred here, do we? Because these are all dominant organisms. They could be heterozygous or homozygous, okay? But we know all of these would be little, right? I know this would be little w, and little g, or whatever for different things. Why? Because the only way we're going to see the recessive allele is if the dominant allele is not around. So that's purebred. Hybrid are organisms with different alleles for a trait. So they're not purebred, they don't have the same. These are called heterozygous. Hetero meaning different. And so that's basically what we're talking about when we talk about being dominant or recessive, purebred or hybrid. We're talking about their alleles, their genes, and how those genes have been passed from their parents. Now, why is this all significant? We've talked about this, and I know this has been some time, but why is this significant? Here's the deal. Mendel actually changed the view of inheritance. Before Mendel, the view was that our genes were kind of like paint. If I take a yellow bucket of paint and a green bucket of paint and I spill it on the floor, what you get is a mixture. You get a blend, okay? And so what scientists and people in general believed is that, hey, I'm a blend of my parents. I get some of the traits from mom and some of the traits from dad, and they kind of blend together and they make me who I am. The problem with the blended genetics is there's one big issue. First of all, if your genetics just continually get blended, then you shouldn't expect to see the same single trait showing up over and over again, because it should get blended and change. And so ultimately, that would lead to more variation. In fact, interestingly enough, the blended philosophy actually fits more with evolution than it does with creation. Creation, okay, the view that God created mankind after his own image, and that we reproduce after our own kind, and that we don't blend traits, okay, that those traits don't change, we're not adding new information. This view actually sees, as Mendel arrived at, that no, what happens is you get inheritance, where these traits as genotypes get passed down. So grandpa and grandma have their son here, and this son gets a little B from grandpa and gets a little B from grandma, and so that son's a purebred. He's like his dad in that he has little b, little b. Well, if he marries someone that's little b, guess what? The only possibility for their daughter is for their daughter to be a purebred, because either little b would still make up for that situation where you'd have little b, little b. So Mendel changed the whole game because his experiments demonstrated that parents' traits don't just blend, okay, that there are dominant and recessive genes. Now, crazy thing about this is during his work in his life, nobody even recognized it. In fact, up until the 1900s, Mendel's work basically was not known about. And fortunately, in the 20th century, his work got the credit that it was due, and that's why he's called the father of genetics. So that is genetics in a nutshell. Please answer the questions that have been throughout the video. I think there's a total of 4 of them. And don't forget to summarize, write your summary on haiku, and I will see you guys later. Bye.